everyone, I'm Alexis Heraclis, and today we'll be talking about chromosomal aberrations and synapses of chromosomes. Before we get started, I'd like for you to pause the video for a quick refresher on the terms we'll need to know. Moving on, how we'll be illustrating today's topics is through a case study of two different individuals, the first being individual 1. Here are individual 1's chromosomes. From what you know, does individual 1 have a chromosomal aberration? If so, what type is it? Pause the video now to figure out your answer. To figure this out, let's first realign the chromosomes so the alleles match up by reversing the sequence of one of them. That's better. Now we can look at what part of the chromosomal sequences are the same and what differs. As you can see, the alleles around the centromere in the center of each chromosome do not match up. Do you know which chromosomal aberration this is? If you said paracentric inversion, you're correct. A paracentric inversion involves a segment of a chromosome that is removed, turned 180 degrees, and reinserted back into the chromosome. Paracentric inversion also has the centromere included in the inverted area, unlike paracentric inversion that does not. Now I pose the question, what would be the resulting chromatids if a crossover occurred between the J and K region? Before we get started, let's use roller coasters as an analogy for chromosomal crossovers. There are many different types of crossovers just as there are roller coasters, which is great for diversity and speciation of cool rides. Although, if these crossovers are not done correctly, it can lead to missing parts and roller coaster failure. Keep this in mind when deciding which chromatids are useful from the crossovers. What you see here is an inversion loop that forms from an inversion during synapses of chromosomes. The drawing shows our two chromosomes from individual 1 and where the potential crossover between the J and K regions will happen, as represented by the X. Now let's figure out the chromatids that result from this crossover. To make it easier to trace the path of the 4 chromatids, I've color-coded each line. Starting at the top and tracing the red strand, you'll see that we get the original sequence. Next is the orange strand involved in the J and K crossover that will follow to produce a new sequence. MIJ, centromere, KLM. Using the same technique for the next 2 strands, and we have our 4 crossover products. Which of these will produce a viable gamete? Using the terms that define a viable gamete, we'll find that both the red and yellow strands are viable because they meet all the requirements. The orange and pink strands are not viable because they don't have all their loci and contain duplications or deletions in their sequence, which is characteristic of paracentric inversions. Now let's look at individual 2's chromosomes and decide if they have a chromosomal aberration. Remember, we first need to realign the chromosomes. From the alignment, we see that they are identical segments, so there's no chromosomal aberration. This also means that there will be no loop present in the chromosomal pathway when looking at the crossover between the J and K regions. Using the same technique as individual 1's crossover, we will get the gamete product shown below. As you can see, they all result in the same sequence, MIJ, centromere, KLHG. They also meet all the requirements of being a viable gamete. Our last question is, between individual 1 or 2, which has the better genetic fitness? The answer is individual 2 because they have more viable gamete products, making them the more reliable roller coaster. Thank you, and that concludes this video.